Good afternoon, everybody. I'm Leonardo Elias. I'm PhD candidate at the Biomedical Engineering Lab, University of São Paulo, here in Brazil. And in this talk, I'm going to present a neuromuscular model that we used to simulate the stretch reflex of the triceps surae muscle. In this figure, you can see a typical recording of the soleus EMG, and you can see that after a sudden stretch of the triceps surae muscle evoked by a toes-up rotation, a first short latency reflex response is observed at approximately 50 milliseconds after the onset of the perturbation. And the short latency reflex, or SLR, is followed by a medium latency reflex response, frequently referred to as MLR or M2. During certain motor behaviors, such as walking, a third long latency response is observed. However, in this talk, we are interested only in the short and medium latency reflex, which are evoked in a seated position with a small contraction or with the subject relaxed. Several experimental studies try to investigate the neurophysiological mechanisms behind each component of the stretch reflex. And the origin of SLR is well established, and it's related to the activity of 1A efferents from muscle spindles. Nonetheless, the MLR is more controversial. Some authors argue that it's generated by the length-dependent type 2 efferents from muscle spindles due to their lower conduction velocities, while others suggest that MLR is generated by a rebound activation of spinal motor neurons evoked by sustained activity of 1A efferents. But why am I talking about a quite neurophysiological phenomenon in a conference on biomechanics? And the reason is that the stretch reflex is used by the neuromuscular system to regulate the joint stiffness. Specifically for the triceps surae muscle, the regulation of ankle joint stiffness is quite useful for motor tests such as posture and locomotion. So, the first objective of this study was to develop a biologically based neuromuscular model with proprioceptive feedback from primary and secondary muscle spindle efferents. By using this model, we've simulated the stretch reflex of the triceps surae muscle so as to investigate the mechanisms behind the genesis of SLR and MLR. This is an overview of the neuromuscular model developed for this study, and the figure shows the connections for a single muscle. Descending commands representing the volitional drive to the spinal motor neurons were represented by 400 independent homogeneous stochastic point processes. The spinal cord included over 1,500 conductance-based motor neuron models divided in 3 motor nuclei commanding the soleus and gastrocnemi, 350 group 2 interneuron models, and over 700,000 synapses. Each muscle was represented by hill-type models, and an EMG model was adopted to represent the global muscle activation. A biologically plausible muscle spindle model parallel to the muscle fibers provide 1A and type 2 efferent activities. The sensory fibers were represented by over 1,500 non-homogeneous stochastic point processes. I won't show the mathematical details here for convenience, but if you're interested, we can talk later or during the Q&A session. And so, each motor neuron model was represented by a two-compartment conductance-based model. We've included sodium fast potassium channels into the soma compartment to generate the action potential, and a slow potassium channel yielding the HP. Synaptic currents were included into the dendritic compartment, and model's parameters varied along the pool. For example, in this graph, you can see the spike threshold as a function of the motor neuron's input conductance. Interneuron models are mathematically more simple. They were represented by a single compartment with spike and AHP generating conductances, as well as synaptic conductances. The spike thresholds of interneurons varied along the pool to represent the scattered recruitment of these cells. As I mentioned earlier, each muscle was represented by hill-type musculotendon units. Passive elements represent the muscle fiber viscoelasticity and tendon elasticity. A mass was included to provide computational stability, and the pination angle varied with the muscle fiber length so as to maintain the thickness of the muscle constant during a given displacement. The contractile element was divided in 2 elements, one representing the slow contracting type 1 muscle fibers and the other the fast contracting type 2 muscle fibers. The force of each element is given by the product of an activation signal and the force length force velocity relations. The activation of type 1 fibers was computed from the spike trains of S-type motor neurons, while the activation of type 2 muscle fibers was computed from the spike trains of FR and FF type motor neurons. Force length velocity surface were reported by a previous study for type 1 and type 2 muscle fibers, and the relationship between musculotendon length and ankle angle, as well as moment arms and ankle angle, were also reported elsewhere. Motor unit action potentials were represented by Hermite-Rodriguez functions, and these mu-ups were widened and scaled depending on its location within the muscle cross-section. The overall muscle EMG is given by the sum of all mu-ups and of all active motor units. This is a quite simple model, but for the purpose of this study is somewhat valid. And finally, we've used the muscle spindle model reported by Milosnich and co-workers. It represents the dynamics of each interfusible muscle fiber, and the primary and secondary afferent activities are nonlinear functions of the tensions produced by each fiber. This is a simulated stretch reflex for the soleus and medial gastrocnemius muscles, and you can see here in the EMG envelope that a short latency response is generated at approximately 50 milliseconds after the onset of the perturbation, and the MLR peak occurs at approximately 80 milliseconds after the onset of the perturbation. The ratio between MLR and SLR amplitudes is equal to 0.5, which is compatible with the experimental data from healthy humans, as you can see in this graph. And here you can see the neuronal spike trains of each element of the model. You can notice that the SLR is generated by the activity of 1A afferents, which excite the motor neurons earlier than group 2 interneurons. However, the MLR is generated by an interplay between both 1A and group 2 activities. Here we have an example showing the difference in reflex response during a small contraction with the muscle relaxed. There is a 10 milliseconds difference in SLR latency and a small reduction in the MLR-SLR ratio. Both results are also similar to experimental data from humans. The muscle fiber length decreases with the stretch reflex, approximately 5% of the pre-stretch value as shown here, and while the joint torque increases. This results in an increase in both muscle and joint stiffness, and in this case the joint stiffness was approximately equal to the value reported by Toften and co-workers. And finally, we have investigated the individual contribution of each pathway to the stretch reflex. As you can notice in the red curve, when the proprioceptive feedback is provided exclusively by 1A afferents, both SLR and MLR response are generated. However, if the 1A afferents are turned off, so that the feedbacks provided exclusively by type 2 afferents, here in blue, only the MLR component of the stretch reflex is generated. So, our simulation results support that the SLR was exclusively generated by the activity of 1A afferents, while the MLR might be generated by both a sustained activity of 1A afferents due to airbound excitation and a lower conduction velocity of group 2 afferents due to the delayed excitation of the motor neurons. Future studies would evaluate effects of fusimotor activity on stretch reflex and joint stiffness, as well as pathological conditions such as Charcot-Marie tooth disease and spasticity after stroke and spinal cord injury. We can also simulate aging with this model. I would like to thank FAPESP for the financial support of this work, and thank you for your kind attention.